So jumping back a little bit to the symptom development in phytoplasma infected palms, here I have an example of on the right, excuse me, on the left, the A image is a queen palm. In the middle is a pygmy date palm, and on the right is our cabbage palm. So in this experiment, what we did was we wanted to quantify the distribution of the phytoplasma in the palm canopy itself. A previous study we did, we had mapped the distribution in the trunk and found that the highest levels were at the base of the trunk and very near the heart tissue or the apical meristem. But we were curious, and that was useful from a sampling and an applied aspect to help guide people in better sampling. But it wasn't terribly relevant for vector work because the insect's going to be feeding on the green foliage of the plant. So we were interested in the distribution in the palm canopy. So what we did was we took leaf samples at different stages of decline from these plants. And the upper left is from the queen palm, the middle one is the cabbage palm, and the lower right is from the Phoenix robellini. And what we found is that we screened all of these leaf tissues with digital PCR, which is substantially more sensitive than qPCR or nested PCR, because we wanted to be sure that we were having the most sensitive approach to see if we could and the more accurate way to quantify the phytoplasma in the leaf tissues. What we found was that the phytoplasma was completely absent from the symptomatic leaves. So you can see some of them that are completely dead. They lacked phytoplasma, at least as far as we could tell with the digital PCR assay. And even the fronds that have some discoloration and are starting to exhibit symptoms are all tested negative for phytoplasma. Now, this is a little bit out of my area of expertise, but when I saw that there was no phytoplasma present in the leaf tissues exhibiting symptoms, and knowing and seeing cross sections of phytoplasma in cells, what I suspected was happening is that the symptoms and death of the palm are a result of physiological stress, where the phytoplasma simply inundates the vascular network at the base of the trunk, which is where we saw the highest levels, and it's basically preventing nutrient flow in the plant, and that stress is ultimately kills the plant. And my other reasoning for this was that typically a stressed plant loses the oldest leaves first, and the more nutrient-heavy tissues such as inflorescences and necrosis. And that's exactly what we see with these infections, is the oldest leaves on the palm die first, and then slowly it gets to the younger leaves. And if you ever see a recently transplanted palm that they've installed in like a business or along a highway, the symptoms you see from the stress of moving and cutting the roots is identical to phytoplasma infections. So the absence of the phytoplasma in the leaf tissues in combination with some of the environmental conditions, it makes me think that the phytoplasma symptoms in palms that decline is a result of physiological stress. But again, like from the previous talk, I was aware that phytoplasmas and other systems produce effector proteins that manipulate the host. I don't know if that's the case, if there's something like that also at play here. But like I said, that's a little bit out of my realm. This was just me kind of playing with ideas in my head. What we did find was that the phytoplasma was present in the spear leaf. So the youngest leaf that's attached directly to the apical meristem is where the phytoplasma resides. So that's a critical piece of knowledge for understanding the disease cycle and how and where and when the insect vector is acquiring the phytoplasma. And in natural settings, we find a very small percentage of the population of insect vector carrying it. It's something like 0.6%. Now, at first, we were concerned that this was a very low percentage for a vector, but when we discovered that the phytoplasma was only in the spear leaf, it kind of made sense, because the time between when the phytoplasma actually invades the spear leaf tissue and the spear leaf is still alive and desirable for the insect vector is a very narrow margin of time, possibly a week or two. So the possibility for acquisition of the phytoplasma is very limited in time and space, which results in a very low percentage of the population carrying the phytoplasma. So the disease we have right now, it's bad in Florida, but it could be a lot worse, assuming all the other fronds had the phytoplasma in them. We use this knowledge to our benefit to refine our vector research in this area. So if, simple logic, common sense, if the phytoplasma is only in the spear leaf, we use that for our acquisition, for our transmission assays. So currently what we're doing is transmission acquisition, establishing acquisition access periods and inoculation access periods using the spear leaves of infected palms. So when we have a palm that's but the other problem is we're still at the mercy of the environment. We can only get a source for the phytoplasma if a palm's infected at the right stage, and we can even reach the infective leaf tissue. Some palms, it's impossible because they're too tall or it's too difficult to get access to it. But if we get a palm at the right stage, we run out, we collect the spear leaf. You can see in this image, we collected the spear leaf, we put it in a container with water, and we stuff cotton around it. Otherwise, the insects crawl down and they drown in the water. So we try to restrict their ability to do that. But basically, my PhD student was allowing them to feed on infective leaf tissue for different periods of time. And we've successfully gotten acquisition of the phytoplasma in Aplaxius crudus. So here's an image of a close-up of the bug feeding on the spear leaf tissue, and an image of the stained salivary glands, just so you can kind of see how big they are and where they're situated. And using the digital PCR, and subsequently we sequenced it because the titer was actually high enough to get a normal PCR, we confirmed the presence of the phytoplasma in the salivary gland. So another degree of confirmation that it's the vector, but we're understanding the finer mechanics of the acquisition. So now we're in the process of inoculating palms, but it's winter right now, so populations of the bug are kind of low, so it's kind of difficult. But we did just find a site in Miami with high population numbers, so we've been going there. So we're moving forward with this research, but the ability of Crudus to transmit it's not surprising, it's not terribly new. What's critical in my mind for this is its implications across the region. And now that we know how to get acquisition of this phytoplasma reliably into the insect, we can start screening all of these other insect species that are closely related to Aplaxius crudus with the other subgroups of phytoplasma. So we can get a better understanding of the evolutionary relationship of these insect vectors. knowing this, again, we study the biology of the insect to optimize our protocols. You can see here the adults of Aplaxius crudus prefer feeding on the underside of the leaves. They like the palms that have the nice fold in the leaf tissue. They like to hide there. So, we don't have a sizable colony yet, so we're at the mercy of the environment, and we go out, we collect cohorts of individuals, expose them to the leaf tissue. Excuse me, and we have our negative control where we don't we test them right away to make sure nothing from that site is carrying the phytoplasma, and these are some of our sampling approaches for collecting the insect. Sweep netting is the most reliable for getting live individuals to do transmission assays. When we're trying to establish distribution and population phenology, we use sticky traps in palm canopies that we can reach. And then kind of an excuse to, get out of the house and go out at night and have a few beers with friends. We set up a light trap and just have fun collecting the adults of Crudus that way. But sweep netting and sticky traps are the most practical. And one of the other important aspects of this, especially with the relationship of Aplaxius crudus to these palm phytoplasmas, is I'm curious and suspicious that similar trends are going to be seen in Africa, and I'm aware of some other taxonomic groups of phytoplasmas that have been isolated from date palms in the Middle East as well as in India. So to me, there's a very, very interesting open niche for investigating palm infecting phytoplasmas in India, Africa, and the Middle East, because I'm suspicious that some of the same trends we're seeing in the Caribbean with regard to symptom progression, distribution in palms, insect diversity on palms, and the vector relationship. My hypothesis is that you would see obviously different species of insect, but I'd be suspicious that it would be within the Succiidae, maybe Durbids, on palms in India or Africa or Middle East, and that there's a similar relationship where you have a high diversity of plant hoppers on palms, and whatever group of phytoplasmas infecting in palms in that region, I would suspect that there's going to be an interesting relationship there. So hopefully some of the findings of what we've documented in palms in the Caribbean, I hope can be useful for especially the spearleaf work on terms of acquisition, can be useful for doing investigations in other regions of the world to look at those other taxonomic groups of phytoplasma. One final thing that we're finding that is interesting is that Aplaxius crudus appears to prefer feeding on infected palms. Now, this image on the left, we published a paper where we used electronic nose technology, which is kind of a crude catch-all volatile detection system. And we found that it could distinguish and give different volatile profiles and signatures between infected palms infected with phytoplasma and healthy palms. So we have the cluster on the right is from infected palms, and on the lower left is healthy palms. Now I'm working with this group of engineers to try and well, they're doing the engineering, not me. I just help with providing samples, confirming infections, and put it in the context of the epidemiology. They do the physics of the electronic nose technology, but we're interested in seeing if we can get better separation. So if we can clearly distinguish the different stages of infection, so that we could potentially use this technology in nursery settings to get detections early, remove plants before they become a risk for further spread of the disease. So that was the first indication that there was a volatile released from infected palms. On the right, this is from a paper we recently published documenting a significantly higher level of insects associated with infected palms. So a combination of a distinct volatile profile using eNose and the fact that the vector is preferentially feeding on infected palms, we're confident that there's a volatile involved, and we're working on identifying that to potentially develop a new management strategy for the vector that's non-insecticide related for palm nurseries, where we can synthesize the compound and use it in a kind of an attract and kill type system in palm nurseries, where, you know, obviously we'll have to do studies to see how many traps we need to shut down the population, and have them preferentially go to our traps rather than the palms. So with that, I would like to thank everyone for your time and attention. You know, it's kind of a rapidly developing field of research. We're finding out lots of new stuff every day. It's quite exciting. So there's always new papers coming out, new research. I have more new insect species to describe than I have time, but it's quite fun. I have all my projects listed up on my website that's here. So with that, I will check to see what questions there are.